Thank you. Okay, I'm Vincent Jemovo from Kenya, and I'm delighted to be here today to present to you about a concept in developing antigen-specific rapid diagnostic test kit using monoclonal antibodies for detection of Tularemia parva. Welcome all. This is the outline of my presentation. I'll just go directly to the introduction, the first part. Tularemia parva is a protozoan parasite that causes East Coast fever in cattle. The disease is prevalent across eastern, central, and southern parts of Africa, especially in Kenya, South Africa, Uganda, Tanzania, and among others in the named areas. This disease is transmitted cyclopropagatively and trastidially by a three-host tick called Rhipicephalus appendiculatus. At least 40 million cattle in the area are estimated to be exposed to the disease. Actually, this is the most fatal disease among cattle in this region. This is projected to result in annual economic loss of over $300 million. You can see how deleterious the disease is and how it is affecting those people living in that region. So I had the idea of coming up with the RDT that can detect this disease. What are RDT? Rapid diagnostic test is a qualitative and semi-quantitative vitro diagnostic medical device that works on the basis of immunochromatographic action like lateral flow or agglutination. They form the antigen-antibody complexes with the specific antigen of the pathogen from the given sample. So my pathogen of interest here is that Tularemia parva. Monoclonal antibodies. What are monoclonal antibodies? They are also called MABs. These are proteins made in vitro that act like proteins called antibodies in vivo. In vitro just means they are made in lab, and in vivo in the body of the organism. As presented in my picture below, you can see how it is produced. This is just an overview where antigen is injected in the model organism. Here is mouse. The mouse will be antenated and will produce the B cells. The B cells will be hybridized with myeloma cells, which forms hybridoma, then results in formation of monoclonal antibodies. Problem statement. About 50% of African livelihood depends on livestock farming, with the main contributing species being bovine. Here in Africa, actually, are facing it. Most African countries are third world countries where we majorly depend on livestock for livelihood. Here in Kenya, mostly in the parts of semi-arid and arid areas, they greatly depend on cattle for livelihood. That's as a source of food, source of income, and the source of the entire dependency in livelihood. East Coast fever is the most important bovine disease in terms of economic losses and restriction of livestock development. Now, this African population depends on bovine, this being cattle. And we have this ECF, the East Coast fever, which is the most deleterious disease that affects this cattle, which provides for this African population. So you can now get how this ECF is deleterious to the economic development among this region. Now, traditional diagnostic methods for Tularemia parva, such as microscopy and molecular techniques like PCR, are time-consuming, require skilled personnel, may not be readily available in resource-limited settings. This has contributed to high rate of mortality and morbidity due to delayed diagnosis and sometimes misdiagnosis. So for a farmer to know that my cattle is suffering from ECF, he or she has to call for the technician and come and pick a sample and then travel back to the lab setting to go and test using PCR or microscopy that require very large time before the results are depicted. And by then we waste a lot of time waiting for the results. This finally results to more death and morbidity in these animals. Justification. The only available diagnostic tests for this disease, the T. parva, are microscopy, immunological tests, and molecular techniques. These are the only available diagnostic tests. And if you can see from yourself, microscopy requires a lot of time. Immunological tests like ELISA and PCR, also they require a lot of time and large laboratory spacing, and also they are very costly. These techniques that are very time consuming, require just laboratory space, expensive and reliable during emergency. Rapid diagnostic tests, it is an answer to this problem. These kits can provide a cost-effective, user-friendly, and quick alternative for screening and diagnosis, especially where quick intervention is required and in remote areas where accessibility to the lab is low. Relevance of the study. How is this study important? Or what do we expect after, in case this study goes through? Number one, we do expect the livelihood of farmers to be protected. The basic food producers in this African world, among of them is livestock. So when we protect this livestock, if you can detect this disease as early as possible, treatment intervention can be taken and thereby saving a large number of animals and hence protecting the livelihood of farmers. Number two, food security. This will be assured to the food security to the whole African population that depends on livestock. Improved disease control, optimized treatment strategies. When the disease is detected as early as possible, the treatment strategies are optimized and hence the health of the animal is catered. It will contribute to poverty reduction efforts in the region. Cattle, mostly in Kenya, ASAL regions, are the source of wealth. So when we protect these cattle, this will contribute to reduction of poverty. It is one measure in which I think we should think and we go towards it for us to reduce this poverty. This is the major disease that we are fighting in Africa, poverty. Then recognition and support of youth involving in research. Mostly, like my research involves a lot of youths, and when this research is recognized, we thereby be promoting the research network among the youths and also inciting them to do more research and thereby improving the health being of the whole world. Objectives. The objectives of this research study is number one, to develop antigen-specific rapid diagnostic test kit. Number two, to stimulate production of monoclonal antibodies. 2, 3, to validate and register the kit. To educate farmers on how to use the kit. To promote networking among the youth by partnering with the various youth groups like GYAN, and also academic advancement by acquiring a master's and a PhD. Hello, can you hear me, everyone?
Yes.
Okay, thank you.
Yes, okay.
Thank you. Methodology of this research study. Okay, this is just a summarized methodology. Number one, we'll begin with sample collection, isolation and purification of these sporozytes, the T. parva sporozytes, production of monoclonal antibodies, test strip preparation, labeling of monoclonal antibodies with gold, nanoparticles, and fluorophores, immobilization and stabilization, quality control, and finally, data analysis. Are you together?
Yeah.
Okay. Principle of this study. This study will be based on the principle whereby my kit will look like this. We'll have the sample application part. This is where our sample, the analyte, will be applied. Then we have the conjugate part, the nitrocellulose membrane part, which contains the test line and the control line. Then the wicking pad. So when you apply your sample at the sample pad, you will follow by applying, it will be followed by applying the buffer. This buffer will carry our analyte up to the conjugate pad and depicted on nitrocellulose membrane. If the antigen of interest is present, it will be captured at the test line, and a bright line will appear together with the control line. So there will be appearance of two lines. This will mark that the antigen that you are looking for is present, and hence the result will be positive. And if there will be appearance of only one line at the control part, it will indicate negative. That's the principle behind it. These are among the references that I used in my study. And also, as you can see, this is the state I'm talking about here in Africa. Poverty is the largest contributor of this third world C in these African countries. This is the cattle that is affected with DCF. Cattle are dying. It's very deleterious. And this picture, another picture, depicts that most African communities depend on livestock for wealth, source of food, and well-being. Thank you for listening to me, and I will be able to welcome you all if you have any question about this presentation. Thank you.